Impact of the microbiome and paralipin-2 overexpression to hepatotoxicin and mutation-induced unfolded protein response activation and lipid droplet accumulation in zebrafish hepatocytes. Stress in the form of alcohol abuse, high-fat diet, and environmental toxicant exposure leads to activation of signaling pathways. This response to unfolded protein accumulation in the endoplasmic reticulum, ER, is critical to cellular homeostasis and mediated by the unfolded protein response, UPR. Chronic UPR induction is maladaptive and known to cause diseases such as diabetes and fatty liver disease. Steatosis is characterized by an accumulation of lipid droplets in hepatocytes. And while this is not pathological, steatosis is a requisite for the hepatic dysfunction and inflammation that occurs in the more severe steatohepatitis. I leveraged well-established models of steatosis in zebrafish to further understand if the microbiome could influence the UPR. Intravital confocal imaging of 5 days post-fertilization zebrafish reared in conventional and germ-free conditions was performed across 3 genetic and pharmacological interventions for lipid droplets and ER. There was no significant impact of the germ-free rearing on ER structure or steatosis severity in stressed hepatocytes. To assess the significance of lipid droplet accumulation and its influence on the UPR, I used a transgenic zebrafish line overexpressing the cytoplasmic lipid droplet protein paralipin-2 in hepatocytes. Lipid droplet accumulation was significantly higher in paralipin-2 zebrafish compared to wild-type controls. Overexpression of paralipin-2 also reduced the incidence of aberrant ER morphology. This supports the conclusion that stabilization of lipid droplets by paralipin-2 protects against ER deformity in the liver, and lipid accumulation could correlate with an adapted response to ER stress.